In this video, we'll talk about the subtypes of T cells. Now, broadly, T cells can be classified into two subgroups. One is CD4 positive T helper cells. Another is CD8 positive cytotoxic T cells. Now, these two types of cells differ on basis of their functions and structures. So the helper T cells help other immune cell types to perform their function. So they coordinate with many cell types. Whereas the cytotoxic T cells are like cold blood killers. They directly kill the cells which is infected by virus, or let's say it's a tumor infected cell. Now mostly T helper cells do their job by secreting cytokines, which are like alarming molecules. They further engage macrophage, dendritic cell and many other immune cells and attract them to fight together. Whereas cytotoxic T cells fight by themselves. They have cytotoxic granules which has enzymes like perforin and granzyme, which lead to apoptosis of the target cell. In this video, we are going to talk about both these cell types, their subtypes, and how it is functionally relevant. So T helper cells are generally produced in the bone marrow. Eventually they mature in the thymus, and at the end of the thymic training, they become naive T helper cells. But these naive T helper cells need to be activated by antigen presenting cells such as a dendritic cell. After the T cell activation, T cell would interact with other cell types, or it will differentiate into different subtypes. Now, antigen presenting cell and T cell interaction is crucial for the activation process. I have a different video which describes the T cell activation process in bit more details. If you want this video, click on the I button. Now, T helper cell, once get activated, can also activate B cells. So it interacts with the B cell and eventually gives rise to the signal that triggers the B cell activation. B cell eventually form the plasma cells. There are many changes happening in the B cells like class switching, isotype switching, and affinity maturation of the antibodies. Eventually, these plasma cells would secrete specific type of antibody and thereby participate in the humoral immune response. So indirectly, Th cell activates B cell as well. Now, there are several subtypes of T helper cells. And with the advancement of technology, there are new and new subtypes coming into picture. So T helper cell can give rise to Th1 type of cells, which play specific role against virus. Now, there are also, that means that particular cell triggers inflammatory response. There are also subtypes known as T regulatory cells, which kind of works like an immune homeostat. It kind of negatively regulates many immune system and prevents the risk of autoimmunity. Both the inflammation promoting T cell subtypes and regulatory T cells are important for our body's immune homeostasis. In a moment, it would be a bit more clear when we talk about some disease, which is a cause of this dysregulation of homeostasis. Now, just to summarize how the T cell activation or subtype specification happens, we have to go back to the bone marrow. Because in the bone marrow, there are hematopoietic pluripotent stem cells which eventually give rise to the T cell precursor. T cell precursor gets into the bloodstream, go to the thymus, and in the thymus, it undergoes very stringent training. And there are positive and negative selection that is happening in the thymus. At any point of time, if you feel like a detailed explanation on these topics, you can click on the I button. Now, after the thymic training, T cells interact with the dendritic cells or any other antigen presenting cells. And this kind of interaction, which happens in the lymph node, helps in T cell subtype specification, or subtype differentiation happens after these kind of specific interactions. So specific cytokines and other chemical molecules trigger this kind of T cell differentiation. So T cell birth, T cell differentiation and T cell lineage commitment are three important processes in the entire immune response. Now let's talk about few of the subtypes: Th1, Th17, Treg and Th2 cells. So the Th1 cells enhances the APC activity, It enhances T cytotoxic cell activation, protects against intracellular pathogen, but it is also responsible for hypersensitivity and autoimmune responses. The key cytokines that are secreted by these cells are interferon gamma and TNF. On the other hand side, Th2 cells secrete cytokines like IL-4, IL-5 and IL-13. Th2 cells participate in protection against extracellular pathogens. Especially, it helps in IgE mediated response and it is involved in allergic responses. T regulatory cell secretes interleukin 10 and TGF beta. It inhibits inflammatory responses, maintains the immune homeostasis. Th17 on the other hand side is also a pro-inflammatory T cell, let's say, because it secretes cytokines like IL-17A, 17F and IL-22. Th17 cells specially protects us against fungal and bacterial infection. Also, it has been implicated in many autoimmune diseases and inflammatory diseases. Now let's talk about how different types of T cells are generated. So it all depends on the types of pathogen that the body encountered, the types of polarizing cytokine that can change the fate, and the master transcription factor that can regulate different gene expression module. Okay, at this point it might not be clear, but let me clarify. This is a Th cell, or this state is known as Th0, so it's a starting point. At this particular point of time, this cell has the possibility to become different terminally differentiated cells such as Th1 cell, Th17, Treg or Th2. Now there are external influence which triggers this terminal fate. The external influences include polarizing cytokine. For example, in order for a Th cell to be differentiated into a T regulatory cell, IL-2 and TGF beta cytokines are important. Whereas interferon gamma and interleukin 12 triggers the differentiation of Th1 subtype. Now all these external influence coordinate with internal signaling modules. There are master transcription factors which triggers this kind of fate. For Th1, the master transcriptional regulator is Tbet. For Th17, it is ROR gamma. For T regulatory cells, it is FOXP3, whereas GATA3 is the master regulator for Th2 subtype of cells. Each of these master regulators trigger specific and unique gene expression modules. And these gene expression modules eventually leads to the terminally differentiated fate. So this kind of give you an idea how different subtypes of T cells are actually generated. So this talks about the T cell differentiation as well. Now, these days, with the advancement of single cell RNA sequencing, there are specific ways by which scientists can understand how different subtypes of cells and subpopulations of cells can be discriminated with the help of transcriptome analysis. Now, let me give you a quick model that how different subtypes of T cell can be formed based on the type of infection. Let's say there is a viral infection in the body. So obviously there are specific TLRs that can recognize viral RNA or genetic material, and that would lead to a specific cytokine production. In this case, the cytokine is IL-12. IL-12 receptors are present in Th0 cells or T helper cells. So there are MHC and TCR related interaction. There is CD80, CD86 and CD28 interactions which are crucial for T cell activation. But alongside that, the polarizing cytokine IL-12 leads to the activation of Tbet gene, or Tbet is the master regulator which is getting upregulated in this process, and this would drive the cellular fate towards Th1 cell. Now in contrast, when there is a helminth infection, there are different receptors or pattern recognition receptors which would recognize this infection. This would ultimately lead to the production of interleukin 4. Interleukin 4 mediated signaling would activate different stats and lead to activation of a different gene such as Tbet. So here we can see that there are different kind of interrelated signaling pathways which can antagonize each other. For example, IL-12 mediated signaling and IL-4 mediated signaling are each important for Th1 cellular fate specification or Th2 cellular fate specification. But each case, the particular modulator of transcription is different, like STAT4 for the Th1 cells, STAT5 or 6 for Th2 cells. The master regulator is different for both these cells. Tbet is the master regulator for Th1 differentiation. GATA3 is the master regulator for Th2's fate. And each of the target genes or the cytokines that they give rise to actually antagonize each other and suppress their fate. So this is kind of like a mutual suppression going on between these two cell fates. Like if a cell becomes Th1, it would do anything to suppress the Th2 fate, and vice versa. That means if a cell gets Th2 fate, it would do anything to suppress the Th1 fate. Now let's talk about Th17 cells, which are generated when naive T cells are activated in presence of IL-6 and TGF-beta. So Th17 cells are really important because they help us or help our body to recognize fungal infections. So when the fungus infection affects us, there are antigen presenting cells which recognize fungal presence with the help of specific pattern recognition receptors that triggers the secretion of IL-6 and TGF-beta that produces Th17 cells. Now the TLR2 is the key key particular pattern recognition receptor against the fungal infection. Now Th17 cells are characterized by their secretion ability of IL-17. Th17 cells are developmentally different from Th1 and Th2 sublineages. So Th cells get polarizing cytokine, upregulates ROR gamma, and thereby become the Th17 subpopulations. Th17 differentiation is controlled by a master transcription factor, which is ROR gamma, that we understood, and this is triggered by the activation of TGF-beta and interleukin 6. Now there are evidences which suggest that ROR gamma is the master regulator, because the mouse which is lacking ROR gamma doesn't have that much of Th17 cells, and they are very susceptible to fungal infection. Now ROR gamma mice have reduced sensitivity towards autoimmune encephalitis. That means it is kind of like resistant towards autoimmune disease. That highlights the importance of this particular T cell subtype in terms of autoimmune disorder. Other autoimmune disorders such as multiple sclerosis, Hashimoto's thyroiditis, asthma, eczema, rheumatoid arthritis, all of these things has to some extent involvement of this Th17 subtype. Th17 cells are dominant inflammatory cells that are present in chronic autoimmune diseases. They produce two major cytokines, interleukin 17, 17F and 17A, and interleukin 22. And each of these cytokines lead to inflammation of the epithelia, such as intestinal epithelia or lung epithelia. Also, these particular T helper cells helps to prevent infections in lungs and in the intestine. Regulatory T cells are the cells that maintains immune homeostasis. So T regulatory cells are also generated from Th cells by the influence of cytokine TGF-beta and interleukin 2. So FOXP3 is the master regulator that gets upregulated, and the T regulatory cell fate is committed. And the key cytokines that are secreted by these cells are TGF-beta and interleukin 10. Treg cells are marked by the lineage marker FOXP3. Now FOXP3 deletion in germline ultimately leads to improper development of the Treg cells. On the other hand, FOXP3 overexpression in other CD4 T cell lineages convert the fate into a T regulatory like situation. That means FOXP3 is really important for the T regulatory cells. Now let's talk about some interplay of Th subtypes in a disease scenario. So we'll take the example of Crohn's disease. Crohn's disease is a disease where a specific part of the intestine gets inflamed. Now in normal situation, there is a balanced level of Th17, Th1 cells versus T regulatory cells. So the pro-inflammatory cytokines and the anti-inflammatory cytokines are kind of balanced. But this balance gets tilted when there is a pathogenic invasion of specific bacteria. And in Crohn's disease, there are more Th17 and Th1 cells compared to T regulatory cells. That leads to huge inflammatory burst in the intestine, and that leads to the Crohn's disease. Now, single cell RNA sequencing has found that there are involvement of Th17 cells and the reduction of T regulatory cells in multiple sclerosis patients. Now let's talk more about the other half of the T cells, the cytotoxic T cells, which are cold blood killers. So cytotoxic T cells have CD8 co-receptors, whereas the T helper cell had CD4 co-receptors. So the question is, who activates cytotoxic T cells? So when a cell is, let's say, infected with virus, then this particular cell expresses of many of these viral protein with the help of class 1 MHC. So many of the viral protein mistakenly gets into the endoplasmic reticulum and get presented on class 1 MHC molecules. Now eventually the class 1 MHC displays this viral peptide which triggers the activation of cytotoxic T lymphocytes. Cytotoxic T cells are loaded with specific cytotoxic granules which has granzyme and perforins. So granzymes and perforins are secreted. These particular cytotoxic vesicles lead to apoptosis. So perforin creates spore in the membrane and granzyme enters the cell. So there are different kind of interactions between a cytotoxic T cell and a target cell. There is fast ligand mediated interaction. There is also perforin granzyme mediated interaction. So once granzyme is inside, it leads to the activation of BID. That leads to release of cytochrome C in the cytoplasm. Cytochrome C can activate APAF1, which ultimately activates the cleaved caspase, or which activates inactive caspase 3 into a cleaved form, which is active form of caspase 3. Caspase 3 is the executor caspase, which leads to apoptosis. On other hand, there are other caspases that are activated by FAS and FAS-L mediated pathway, but the downstream trigger point is same. These caspases work in a cascade to activate the executor caspase or the caspase 3. And ultimately, that triggers the events of apoptosis. Ultimately, the entire cell dies, which is infected by the virus. So the virus gets eliminated in this way. Now, these cells are really important for tumor fight against the tumor. But before that, let me tell you that there is another kind of cells known as memory T cells. Memory T cells are generally long-lived, quiescent, but they kind of remember the incidence of pathogen exposure, as if they have a complete memory of the history about the pathogen. In order to understand memory T cell, we have to understand the phases of T cell mediated immune response. First, there is a recognition phase, then there is a clonal expansion phase where the T cells or a subtype of T cells expand in a clonal fashion. Then there is a activation phase where the T cell is activated or where it gives rise to plasma cell activation. And eventually there is an effector phase where the cells are destroyed or the pathogen is eliminated. After the elimination of pathogen, so many effector T cells are not required. So they get selectively eliminated, and this is known as a declining phase. Eventually, many of these effector T cells would die out of apoptosis, but some of them would remain. And this remaining one would become memory T cells. Next time when there is an infection, these cells would show an augmented response. So let's see why T cells are necessary. Generally, after all the infection or the pathogen is eliminated, many of these cells are dying and some of them are becoming T cells. Now T cells, some of these are becoming memory T cells. Memory T cells and naive T cells are not so different. So the memory T cells and naive cells both are in quiescence or G0 phase of the cell cycle. But the memory cells have a tendency, or they have a less stringent reactivation criteria. So they can get back to the cell cycle and proliferate rapidly when it is necessary. So the naive T cell cannot do that very quickly until and unless it is activated by the antigen presenting cell. But naive memory T cell can quickly get activated with the help of many of these cell types such as macrophages, B cells, or many other antigen presenting cells. So reactivation is quite a quick process. Now naive T cells and memory T cells are very different based on the expression of distinct receptors, co-stimulatory molecules present on these cell types, such as CD44, CD62 ligand, CCR7 chemokine receptors, etc. Now T memory cell display the property of recirculation. So memory T cells are of broadly two types: effector memory T cells and central memory T cells. In this video, we are not going to talk about these subtypes in details. If you want to know more about these cell types, you can watch a different video. Now let's talk about the possible models of memory T cell generation. So naive T cells eventually go through a effector phase, and once the effector phase is done, it becomes a memory T cell. Another hypothesis says that at each substage, there is a possibility of differentiating into memory cells. So the trajectory is defined earlier, but we don't have a clear-cut clue about this model, so still it is debatable. Now let's talk about exhausted population of CD8 positive T cells. As the name suggests, these are some T cells which are highly exhausted. So we'll talk about what are exhausted T cells, when they are generated, subtypes of exhausted T cell, and why they are important clinically. Exhausted T cells are literally CD8 positive T cells which are very tired. Their surface expression is very different from a normal CD8 cytotoxic T cells, and generally they are defined by the molecular feature of TCF negative, EOMS high, and Tibet low. They have reduced proliferation capacity, and their cytokine secretion capability is much less compared to normal cytotoxic T cell. So how these exhaustion takes place? It all depends on the magnitude and the duration of the overall T cell response. So in general, after a T cell response, after antigen exposure, there is a T cell response where there is a naive stage, there is an effector stage, and there is a memory formation stage. So this is happening in the normal scenario. But when the antigen exposure is chronic, happening over a long period of time, or let's say there is a tumor formation which is staying there for a long time, in those cases, the naive T cells get activated into the effector T cells. But eventually they get exhausted, and their proliferative capacity, cytokine secreting capacity gets reduced, and this state is known as exhausted T cells. Now exhausted T cells are really important for cancer. Like generally these are the cells which eliminate tumor. So but the tumor cells are also growing at a rapid rate. So after a point of time, the tumor cells have grown in so much in number that the T cells are kind of exhausted to fight them. Now these T cells have eventually become the exhausted cells. Sometimes these T cells has no other options other than becoming the cytotoxic exhausted T cells. These cytotoxic exhausted T cells are hypofunctional. Their efficacy has decreased, and this is a major challenge when it comes to T cell therapies. Now this is transient state, but it is not good for the overall body's response. But it's kind of like an obligation. When exhausted, if the T cell continues to fight back, it has to die. But another option is to work in a suboptimal fate and survive. Because exhausted T cell state is a reversible state. Now there are many negative regulatory modulators which triggers this kind of exhausted T cell state, like PD1, PD ligand interactions, CTLA interactions. This lead to this kind of exhaustion. There are also other receptors like LAG3, TIM3, which lead to reduction in proliferation and promoting a state of kind of exhaustion. Now there are so few factors govern this kind of situation: persistent antigen presentation, nature of infection, and then the negative co-stimulatory signals. But let's talk about why it is so important in case of cancer. Now let's say you want the body's immune cell to eliminate tumor and kill the tumors. Now after a point of time, if these cells are exhausted, that's not a desirable situation for your body, right? So these days, people understood that negative co-stimulation is a key mechanism to drive exhaustion. So in that case, many cancer cells upregulate the inhibitory co-stimulatory receptors, and that reduces the potential of the T cell therapies. That's why these days people use antibody mediated therapies where they block the PD ligand or many other co-stimulatory ligands such that this inhibitory co-stimulatory action cannot be triggered, and cytokines can be generated and tumor cells can effectively be eliminated. So these are very popular therapies these days. So I hope this was useful. If you like this video, give it a quick thumbs up. Don't forget to like, share and subscribe. You can support us using super thanks. See you in next video.